Thank you so much for coming, everyone. It's great to see so many people interested in Riverflies. So like Eileen said, my name's Laura. I'm the Community Woodland Ranger at Newbattle Abbey College. And in a couple of slides' time, you'll hear from Rebecca, who's our voluntary project coordinator as well. So obviously, we're going to be talking about Riverfly on the Esk, as you can see from our slides. We're going to be explaining about the project, including information on what we go outside and record, including the various Riverfly groups. We're going to talk about who's involved in the project, the importance of these surveys, particularly in the context of the South Esk River, and how we hope that the project may grow in the future. Next slide, please, Becky. Perfect. So firstly, key facts about the project. Who, what, where, when, and why. Next slide. So firstly, we've got who. The group was formed back in 2019 as part of a community engagement project run by Newbattle Abbey College. And there's always been a keen local interest in the river's water quality following regular pollution incidents that happen along the river. And this motivation really helped drive forward the formation of the group because so many people were interested in how they could help and get involved. So an official training session was organised and run by SEPA for April 2019, and we had 12 local people come along to that, which is really great. And you can see here in the picture, someone was demonstrating the sampling method, and then everyone got to go in and have a go. And then we identified all the different creatures we found, which was really good. Since then, we've recruited more potential volunteers onto a waiting list. Obviously, COVID's put a bit of a halt on our training sessions for the moment, but we're hoping to be able to organise another training session like this and get people ready to go out and survey. So hopefully once lockdown eases, we can get on with that. Next slide, please. Brilliant. So now we're on to what. So the group is part of a nationwide community-led citizen science project called the Anglers Riverfly Monitoring Initiative, or ARMI for short. And that collects aquatic invertebrate data on waterways throughout the UK. So it's not just a local thing, it's a national network, which is really cool. The project relies on volunteers from all different backgrounds coming together and surveying rivers for specific Riverfly families, which we'll tell you about later on. So the presence or absence of these groups, as well as the relative abundance of each group, then translates to a biological index score, which allows volunteers to monitor the water quality of the river. And then if this score falls below a designated threshold that we've got for each site, the relevant environmental authority is notified, and then they come out and look at why that might have happened. So it's all kind of linked together with SEPA and environmental protection agencies, which is really good too. Next slide, please. So where, at the moment, we've got 3 survey sites along the River South Esk. 2 are located in the grounds of Newbattle Abbey College, 1 upstream of a known combined sewer overflow, or CSO, and 1 is just downstream of that. So we're hoping to be able to monitor water quality given the CSO being there. And then our last survey site is just at the Sun Inn in Midlothian, and again, it's just downstream of a CSO. Next slide, please. Thanks. So when surveys are completed on a monthly basis as a minimum requirement, and usually we try and survey throughout the year. So exceptionally bad weather can put a bit of a pause on surveys sometimes, but we have been out in the snow before. I'm not always there, but the volunteers are a really hardy bunch and they power on no matter what the weather. Next slide, please. So why? This is a really important thing to talk about, is why we go out and do these surveys. So the initial driver behind setting up the group was the enthusiasm of the local community to do something to help protect the river, like I said, from pollution and conserve important aquatic habitats. The main aim of the surveys is to highlight any pollution incidences that do occur, and this would obviously be picked up as a significant decline in biodiversity of these riverfly groups. However, there are also a number of secondary benefits, including positively engaging the local community in nature, And obviously with lockdown easing, that's really important with getting people outside and connected back to the natural world. It also provides opportunities for education, training, and volunteering to communities. And also it gives people a chance to get to know people in the local area. So lots of our volunteers have met each other through the group and they get on really well. So it's nice to see like a little core forming, and they're really friendly and nice and chatty. So we welcome anyone new to the group if you're keen to get involved. Next slide, please. So partnerships. Partnerships are at the heart of this project and allow it to have such a wide-reaching impact. As we've discussed, the Riverfly on the Esk group is part of a national network of community-led volunteer groups that collect data for the ARMY project. But ARMY is just one of the projects that's coordinated by the overarching Riverfly partnership group. The partnership is made up of organisations, landowners, local authorities, volunteers, and scientists working together to meet three main aims. The first aim is to further the understanding of riverfly populations. The second aim, to protect river water quality, and the third, to conserve river habitats. So overall, the partnership is hosted by the Freshwater Biological Association. Next slide. In terms of data, the ARMY project involves recording 8 commonly found freshwater invertebrates, which we're going to talk about on the next slide. But as you can see from this map here from the Riverfly Partnership website, there's a huge spread of Riverfly survey groups across the UK. In Scotland, the majority of groups are situated around the central belt that you can see there, and there's only a few up north. So hopefully as the years kind of continue, more groups will get formed and set up. Next slide. So finally, we're on to what are riverflies? I can hear you all asking it in your brains. Riverflies are aquatic invertebrates found in rivers. They spend most of their lives as larvae, emerging as short-lived river adult flies. So the most characteristic one you might have seen are the mayflies that you get as the weather warms up along the river surface. They act as biological indicator species for freshwater ecosystems, meaning that if environmental conditions are poor, they are most likely to be affected and shown signs of stress. So there are 8 target groups that we look for on the surveys, and they're from 3 key areas. And these are recorded in the ARMY surveys. So we've got 4 upwing flies, 2 caddisflies, 1 stonefly, and 1 gammarus. And those are the 8 that we make sure that we record. But obviously, if we spot something else interesting, we record that too. But it's just those 8 species that are used to make that biological index score. And now I'm going to hand over to Becky, and she's going to continue with the presentation.
Can you hear me? Yeah, figured out how to unmute myself. That's always a good start. Hello, my name is Rebecca. I'm an ecologist and live locally in Midlothian, and I've had the absolute joy of being able to coordinate the Riverfly on the Esk Project. So I'm just going to I'm just going to talk through the practicalities of this and how we go about surveying and getting the samples. So one of the issues, and it's not only an issue for the citizen science project for determining cause and effect of any ecological project, is variables. So there's a variety of contributing factors that can muddy the waters when you're collecting data, and muddy the waters quite literally in this case. So what we have to do is to try and control these as much as possible through experimental design. And luckily, this has been done for us. So the survey methods were developed by the Riverfly Partnership, and it's a simplified version of a routine biomonitoring method used by regulatory agencies within the UK. And it's delivered through a training programme, through standardised technique and standardised kit. So the result is an increase in the data accuracy that we get, and it also provides an easy to use method for citizen science. So how do we do this? So we do this through a 3-minute kick sampling. So each river has a distinctive anatomy, if you like. as each is composed of a series of geomorphic units, which could be pools or riffles or runs within the river channel. The distinguishing abiotic conditions of these units results in specialized environments that are known as microhabitats. So the abiotic conditions, which could be dissolved oxygen or turbidity, light, temperature of these microhabitats, can then influence which aquatic species can survive and reproduce in any given location and time. So the 3 minutes of kick sampling in total are divided up into sampling points across the river to try and get then a representative picture of that river habitat. And keeping in mind at all times, and it's very important of working with rivers, it's a dangerous environment, so you're keeping in mind all times the safety and access of the site suitability. So the kit that we use is, again, a standardized kit. So it's a standard sampling net with a 250mm frame, 500mm deep net bag with a 1mm mesh. So sampling trays, gloves, buckets, sorting trays, flask of coffees, plastic spoons, whatever else you take along. But the standardized kit can be purchased and is used across all the Riverfly partnership sampling sites. So the technique is to place the net in the water at the chosen site and put it flush with the bed of the stream. You then stand upstream and disturb the area in front of the net for a given period of time. Once you've done your 3-minute kick sampling across the stream, this is then followed by a 1-minute rock check. So you spend 1 minute going through with your sampling net, selecting larger stones that you may not have been able to disturb through the kick sampling, but will provide a different habitat and may have different invertebrates hiding underneath. So that you then wipe them into the mouth of the net to dislodge the organisms which may have been collected into the sample. So now you have your collected sample in hand. So you then take it to the shore and the side of the river, and you'll empty it out into your sampling trays, which is a picture you can see here on the right. Let the count begin. So as Laura has talked through, we're focusing on 8 target groups. So you see the picture on the left-hand side. We're looking for these 8 groups. Now, it might be a little bit intimidating. You can wash in a really large sample and things are just running around a mass. So there's techniques that you can use to help with that. So what we tend to do is divide the tray into sections like a grid, and we'll all huddle round and we'll sort of take our different sections, and then we'll focus on each target group and then do an estimated count of each of these groups. What we like to do, and it's been a choice for ours, is to just go through. There's so many interesting things in these trays. So we'll just note down other things that we see as we go along. And some things you just don't want to find. You'll see there in the second picture, we've got a very large, and I remember well, very smelly lump of wet wipe, which was caught in our sampling net. And sometimes the samples themselves will make your eyes water. But yeah, that's the downside of the pollution in our rivers at the moment, unfortunately. So in the centre here, you can see the sorting trays. Now, what we do is we have a bit of fun with these, so we can pick out the interesting invertebrates that we find, and also our 8 focus groups. And it just allows you to have a closer look at them, see them moving around within the trays. And if you've got a hand lens, it's your chance to get the hand lens out and have a closer look. And we have a nice little crop of the cased caddis in the photo next to that. And then the one in the end is one of our favourites. So this is the burrowing mayfly, which is absolutely beautiful markings over it. So I couldn't help myself but popping that one in. Unfortunately, we've only found a few of these during our sampling. They require quite good oxygen levels within the water, so a good healthy environment. But anyway, there's a little bit of a party when we do. So as I said, we can often find some real weird and wonderful things, and this was an absolutely fantastic specimen, and hopefully this is going to play. Aha, there we are. Beautiful and slightly freaky all at the same time. So this is a hairworm that our sampling crew had found in site number 2. Just let you enjoy that for a wee minute. And the next one, I remember looking in this tray and thinking, oh, where is everyone? And lo and behold, our gummerists were hiding in what Laura informed me as a beach cone. So there was quite a few of them and they were packed full of gummerists. It's also a good opportunity when you're looking at your trays to keep an eye out for invasive non-native species, because they can have catastrophic effects on our native populations. So it's worthwhile spending the time having a look and noting down where and when you find them. So down to recording is, after all, part of the reason why we're there. And here is an example of a field recording form that you can download from the Riverfly Partnership website. And this is the moment where I normally hear the footsteps in the door slam, but it's not as intimidating as it looks. It's got all the relevant information you can see along the top. You write down information about the site and the grid reference, and also your trigger level, who else is along with you. You've got the date. Down on the left-hand side, you can see where you list the different groups separated into your 4 mayflies, your caddisflies, stoneflies, freshwater shrimps, and also a section for extra notes. And through the highlighted columns there, you can see the one on the left is your estimated number, and on the right is a score that's generated from that estimate. And if you look down at the table on the bottom, you can see how your scores are generated. So that's the nice field recording form with all the information that you need. And here is Riverfly on the Esk's very own field notebook. Anyone who works in the field will be familiar with the slightly damaged, slightly soggy, smudged, but very valuable notebook. So this comes with us through every sampling site. So this data can then be downloaded onto the Riverfly Partnership Army data section, which is really straightforward once you're in the rhythm of it. So I'm not going to linger too much on this because it's the raw data. So what you can see here is the 3 sites, sites 1, 2, and 3 that we sample, and the line graph represents the different colours represent the different groups. So you can see them wiggling across the graph there with peaks and troughs through the different months that we sample. What's also clear here is a complete break in the data for all 3, and that is, I'm afraid, when the world stopped for COVID, and so did we. So there was no sampling through these months, which we were all very disappointed about. But that's what the raw data looks like, and it's just a bit of fun to see it. And these are the graphs that are generated through the Riverfly Partnership website, and they give us our Army scores for each of the sites. So the Army score for each of the site is generated by allocating a score of 1 to 4 according to the log 10 abundance category for each of the target taxon, and summing the scores of all the target groups. So the scores, you remember, were from the data sheet in the bottom, the small table at the bottom, the generated scores. So the local regulatory authorities give each of the sampling sites a trigger level score based on the long-term data for the site. So you can see along the red line at the bottom is the trigger score that has been put on for our site, and we're assuming at this stage that this is temporary and are waiting to be assigned our actual site threshold. So the grey line along the bottom, which is your Army scores, so the higher the Army score, the better the quality of the river. And the trigger level is set quite far below this Army score. So as the grey line tracks along, the red line along the bottom, but if the grey line was to go below the red line, that would trigger a response. So what we then have to do is go back and it would resample the site. So what it suggests is that it's perhaps been a serious pollution event, which has caused the abundance or diversity to dip below that trigger level. So once we've gone out and resampled the site, we then inform the local agency officer, who will come up and investigate and hopefully then take the appropriate action. So in addition to detecting pollution, what's interesting is this kind of citizen science sampling could also look at show before and after responses to any sort of river restoration. So if you had an area that you were restoring, perhaps you're doing riparian planting, you'll be sampling before, and you can continue sampling to see the effects of that. So it's quite a neat system, simple but can tell you a lot. So now we've come on to the P word, which is yes, pollution, which is causing a lot of problems and to a lot of our rivers. Less than half of our rivers have a good or high environmental status. And there are many reasons for this, but unfortunately, most of them are caused by us humans. So we have to ask ourselves for each of the sites, what's entering the water? How is it entering the water? How is it likely to impact the waterway according to the anatomy of both the water body and the surrounding areas? And then what's looming over us at the moment is the pressure cooker of climate change, which is then going to put added pressure on all of these other impacts. The Water Framework Directive, which states that most of our water bodies should reach a good ecological status by 2027. So we're all crossing our fingers and toes for that one. But given that Scotland's got over 125,000 kilometres of waterway, studying large-scale patterns in nature requires vast amounts of data to be collected across an array of locations and habitats over spans of years and even decades. So one way to get this data is through projects like this, through citizen science. And you know, it's not just the data, it's not the only value that it brings. Citizen science results in a sort of permeation of learning into the consciousness of communities, so increasing the will of people to act. And more simply, we can't save what we don't know it's there. So it's really, really important for everybody to get out and look. And trust me, we do have a lot of fun with that. So, you know, the time for action is now. It's not too late to do something about it. So, yes, I wasn't keen on saying the word poo on screen, but yes, you just have to remember that the only thing that should ever go down your toilet is pee, poo, or paper. And that's something that we can all do on a daily basis. Let's keep that in mind. So to the future, this project has been absolutely amazing to work with, and I'm really grateful for Laura starting it up and giving me the opportunity, and that we've got a really great bunch of volunteers that, yeah, we've been out in the rain, we've been in the snow, and we still do it with a smile on our faces. And yeah, there's heaps to learn about the river. So we want to keep on going. So our plan at the moment, we have roughly about 20 people waiting to be trained. And so we're trying to recruit and train more volunteers all of the time. And obviously at the moment, it's a bit tricky with COVID, but we're looking at online ways of doing this. So we'd also like to expand. We're currently focused on the south branch of the Esk, but we've got really interested groups of people both in the north and the Lothian branch who are keen to get involved. Yeah, it's really important to work with local organisations, you know, sharing the data, sharing what we're doing, making sure that we're not overlapping areas, that we're sharing out such a large area of river, so we can cover as much as we can and learn as much as we can. Water testing is something our group have also been interested in looking at, which I'm currently in the process of trying to sort that out. And developing our training, you know, restricting the number of invertebrates to target groups in the basic army protocols to easily recognisable taxa helps ensure that consistent data is generated, even from novice volunteers. However, this taxonomic restriction means that the army may be sensitive only to major pollution incidences that remove a large part of the macroinvertebrate assemblage of the river. So to provide a more kind of nuanced assessment, it's necessary to add more invertebrate groups to the Army protocol. So, you know, this is what we'd like to do. We'd like to learn more, but we're being guided really by what the volunteers would like and what we're trying to achieve. At the moment, we have got our group application in to be registered as a charity, so that way we can get more kit and repair more kit. And I hear there's calls for gazebos and coffee bars to take out with us, so, you know, that would be nice. So these are a couple of the initiatives that we are looking at. Obviously, the Extended Riverfly Initiative and the Smart Rivers Programme. And basically, they'll both build on our knowledge of the river and help us learn more and contribute more.
Rebecca, just to say that's coming up for about 25 minutes, but take as much time as you want, okay?
Well, that's absolutely perfect, because we're on the penultimate slide, and that's just to say thank you for everybody who's supported us. And join us. We would absolutely love to hear from you, whether you're local or further away, whether you want to start your own group or join our group, whether you want to take away invasive plants from the rivers we're working on. We just like to work together. It's the best way to get the job done. So that's both Laura and myself, our emails are there. And thank you for listening, and any questions. Thanks very much.